In the name of God, today I want to talk about the second week of development. In this week we have a formation of the bilaminar embryonic disc. During the second week we have some events. The most important thing is differentiation of the inner cell mass or embryoblast in two layers, epiblast layer and hypoblast layer, and we say that these two layers together making a bilaminar embryonic disc. In the more development, bilaminar embryonic disc give ectoderm, mesoderm and endoderm layers of the embryo. I will talk about in the next session by details. And also the amnion cavity, amnion fluid, yolk sac, connecting stalk and chorion formed during the second week. Now I started day by day some more important events during the second week. First of all, as you remember, in the end of the first week, the embryo or blastocyst attached to the endometrioms by receptors, and this time gradually the inner cell mass differentiated in two layers. The yellow layer is hypoblast, is a columnar layer, and epiblast at the columnar layer, the hypoblast is cuboid and epiblast is columnar layer. The blue color here is epiblast, the yellow color is hypoblast. And this times the outer cell mass or trophoblast making the two layers. The first layer that made by the outer cell mass or trophoblast is named is cytotrophoblast, and the cytotrophoblast doing mitotic divisions and making another layer that name is syncytiotrophoblast or syncytium. The one as most important characteristic of the syncytium is a loss of the boundary between the cell, and as you see here we don't have any border between the cells, and finally making a multinuclear layer without any boundary between the cells. This syncytiotrophoblast is very important because these layers or these cells secreted one specific proteolytic enzyme that name is matrix metalloprotease. This enzyme break down extracellular matrix around the cells, and as a result the embryo doing more penetration and more implantations. And also the syncytium allow passage of blastocysts in endometrial wall. This layer totally surround the blastocyst, secrete one important hormone that name is human chorionic gonadotropin or HCG. This is a good marker for the diagnosis and presence in the urinary pregnant woman, and it's a marker for the diagnosis of pregnancy. This HCG also maintains decidua and corpus luteum. I will talk about the decidua. Decidua is some part of the endometrium, and this decidua is important for the formation of the placenta. And also syncytium generates space that filled by maternal blood. These spaces is name is lacuna. I will talk about it. This is a role of the syncytiotrophoblast. In this picture you can see that the real human blastocyst This is a endometrium, uterine endometrium, the embryo, epiblast, the columnar layer, and cuboidal layer, the hypoblast layer, the cytotrophoblast, and this one that has a white spaces. These white spaces is a syncytial trophoblast, and the name of this white cavity between the syncytium cell is name is a lacuna. By 8 days, gradually some cells proliferated and differentiated from the epiblast layer and making a new flat cells that name is amnioblast. As the result of the formation of the amnioblast, first of all, small cavity is made that name is amnion cavity. In this cavity, the floor made by epiblast cells and roof is made by this one, the floor made by epiblast and roof made by amnioblast cells, and the first cavity that's formed is named is a amnion cavity. And here you can see that the more penetration of the endometrium by embryo, and you can see that the more differentiation in the syncytial layer. You can see here we have a multinuclear layer without any boundary between the cells. This is a nucleus of syncytial cells, and this is a cytotrophoblast layer. In the next day, by 9th days, you can see that the embryo completely coming to the endometrial stroma, and the site of implantation closed by spatial tissue that's secreted by syncytium. The name of this barrier is forming here is a fibrin coagulums. This is a temporary tissue. And the next important things that happen in the 9th day is the formation of the new cell layer that name is Hooser membrane. These cells, some references say that originated from the hypoblast layer. You can see that the pathway of the generation of these layers, and when this layer is completed, the new cavity is formed that name is a primary yolk sac. Now embryo has two cavity, the amnion cavity and yolk sac, primary yolk sac. But the most important things or most important events that happens during the 9 days is formation of the small cavity in the syncytium layer that name is a lacuna. As a result, the 9th day is famous to lacunar stage. In the next day, around between the 10 to 11 days, as you see in here, a cellular extra embryonic reticulums, attention please, a cellular layers form between the Hooser membrane, this is a Hooser membrane, and cytotrophoblast, because before the formation of this extra embryonic reticulum, the Hooser membrane has direct attached to the syncytium. In the next day, it means in 12 days, a new cell layer is formed from the epiblast layer. Here this is the epiblast, and this layer, this red cells originated from the epiblast and making a new layer between the Hooser membrane and cytotrophoblast layer. The name of this new form layer is extra-embryonic mesoderms. Gradually, some spaces, small spaces, is established in the extra-embryonic mesoderms, and when these small spaces join together, making a large cavity, as a result, extra-embryonic mesoderm, here the red colors, divided by this cavity in two layers. That layer that covered directly and situated over the yolk sac is named is somatic splanchnic layer, and that layer that cover inner surface of cytotrophoblast, this is a cytotrophoblast, and this red color cells is somatic layer of extra-embryonic mesoderm. This layer not only cover the undersurface of the cytotrophoblast, but also cover the roof of the amnion cavity. In conclusion, by formation of the chorionic cavity or exocelomic cavity between extra-embryonic mesoderm cells, the extra-embryonic mesoderm cells divided two layers. One layer that cover the yolk sac is named as splanchnic layer, and one layer that cover on the surface of the cytotrophoblast cells and roof of the yolk sac is named as somatic layer of extra-embryonic mesoderms. But the most important event in the day of the 12th is a first communication between the maternal capillary system that here named is sinusoids. These sinusoids drain blood from the mother capillary into the lacuna that form in the 9 days. This is the first communication between the mothers and embryo blood system. As a result, these days is famous to the utero-placenta circulations. Here again, real embryo in 12 days, you can see that primitive yolk sac. This is a Husserl membrane. Here is extra-embryonic mesoderms. Here, this is a cytotrophoblast layers and epiblast, hypoblast. The columnar layer is epiblast and cuboidal layer is hypoblast. Here is not show by detail about the somatic and splanchnic layer of the extra-embryonic mesoderms. Here you can see that another days around here you can see that extra-embryonic mesoderm formations. And what about the next day, around the 13 days, a new layer, here you can see that, is formed by hypoblast cells. It means that we have a next proliferation in the hypoblast layer, and this hypoblast proliferation making a new membrane. Here show that a new membrane is formed by the proliferation of hypoblast cells. This new membrane migrate out over the inside of the extra-embryonic mesoderms. Here, this is a extra-embryonic mesoderms, pushing the primitive yolk sac. This is a primitive yolk sac, pushing the primitive yolk sac in front of it. These new layers become endodermal lining of definitive or secondary yolk sac. It means that in these days, a new yolk sac is formed from the hypoblast cells, and the name of this new cavity is secondary or definitive yolk sac. As a result, the former yolk sac or primitive yolk sac pushed to the chorionic cavity and making a temporary and transient structure that name is primitive yolk sac cyst or chorionic cyst, and gradually this cyst is disappear. As a result of the formation of the secondary yolk sac, the chorionic cavity be larger because this definitive yolk sac or secondary is smaller than primary yolk sac. Again, the embryo around 13 days, you can see that yolk sac, amnion cavity, and epiblast, and this large cavity is chorionic cavity. Here we have a somatic layer of extraembryonic mesoderm and cytotrophoblast layers. In the final development, in the 13 days, you can see that remnant of primary or primitive yolk sac as a name of exocelomic cyst. And another important things that's happened during this day is complete obliteration and repair of the implantation site, because as you remember, here in the site of implantation we have a temporary tissue as a name of fibrin coagulum. In the 13 days, the new epithelial layer displays to fibrin coagulum and the site of implantation completely repaired. And during this repair we have a small bleeding in the site of the implantation, and maybe this be confused with the bleeding as a result of the menstrual cycle, but this bleeding, amount of this bleeding is less than the bleeding in the time of the menstrual cycles. This bleeding is caused by implantations. At these times, in the 13 days, formation of the placenta is started by formation of primary villi and the secondary villi, and I will talk about the formation of the villi and the role of the villi for the formation of the placenta in next session. This is a summary of the second week. You can see that the epiblast, hypoblast around 7 days, cytotrophoblast and syncytium layers. In the 12th day you can see that the formation of the primitive yolk sac, cytotrophoblast, syncytium, and here you can see that the communication between the maternal sinusoid with lacuna. And as you remember, we say that this is the first time for the communication, and the 12 days is famous to uteroplacenta circulation. Approximately 15 days you can see that remnant of the primitive yolk sac as a name of the exocelomic cyst, and here the yellow color is yolk sac. Here the secondary or definitive yolk sac, the roof of the yolk sac made by hypoblast. Here is a amnion cavity, and floor of this cavity is formed by columnar layer. That name is a epiblast cell layer. Sometimes we have some abnormality during the formation of the embryo. One of the important things that maybe it's happened during the implantation is formation of the hydatiform mole. We have two kind of the hydatiform moles. One of them is partial hydatiform mole. As you see in the picture, is produced when a normal oocytes fertilized by two sperms. As a result, we have three pronuclei. One of them, the red color, is come from the mother, and the blue color it's come from the fathers. And as a result, we have a triploid embryo here, because we have a 3 pronuclei. Another kind of the hydatiform mold, its name is a complete hydatiform mold. This kind of mold is are produced by 2 mechanisms. One of them is when an oocyte is inseminated by single sperms, like a normal situation, but the female pronucleus is lost. As a result, we have a single male pronucleus. This single male pronucleus divides to form 2 diploid nuclei, which combine to form a diploid nucleus. But here we have only the paternal genomes. As a result, only the placenta is formed in this embryo. In another way, to formation of the complete hydatiform is happen when one oocyte is inseminated by 2 sperm and female pronucleus is lost. Again, we have 2 pronuclei that both of them are come from the fathers. Again, here we have a paternal genomes. Here, these pictures, you can see that the histological slides that show that clear swollen really characteristic of these structures. In this patient, it means that in hydatiform molds we have a high secretion of the HCG, and the patients show that the sign of the pregnancy, but unfortunately only the placenta is formed, the embryo isn't formed. Sometimes the embryo is not implanted in the normal site because usually the embryo is implanted in posterior superior wall of the uterine cavity here, but maybe implanted inside of uterus in abnormal position or outside of the uterine. The inside of uterus, uterine corpus, or may be implanted in the internal opening of the cervix as a name of the as a result of this kind of implantation. We have a kind of the placenta, his name is placenta previa, is formed. But the most important site for the implantation is a implantation of the embryo in the ampullary region of the uterine tubes. That site that normally fertilization happens. The embryo cannot move toward the uterine cavity and remain in this area. These implantations continue to more developments, and the pregnancy continue till end of the 2nd month of the pregnancy. But after the 2nd month, as a result of the rupture of the uterine cavity, the embryo is aborted and the patients or pregnant women have a severe bleeding, and this is urgent. At another site, for the ectopic pregnancy, on the surface of the ovary, on the surface of the mesentery, on the abdomen, and another site of the pelvic. Here you can see that the ectopic pregnancy in the uterine tubes, in the real embryo. And finally, here you can see that 2 questions for more review, and please answer this question. And this is like my final exam questions. And now we have one conclusion about the second week. The second week of development is known as the week of tubes. What it means? It means that everything that form in this weeks has two layers. For example, the trophoblast differentiated in two layers. That name is cytotrophoblast and syncytial trophoblast. The embryoblast form two layers, epiblast and hypoblast. Extra-embryonic mesoderm split into two layers, somatic and splanchnic layer. Two cavity form, amnion and yolk sac cavity, and two yolk sac is formed, the primitive and secondary. That's all. This is the second week of the development. In next session I will talk about the third week of development as a name of gastrulations stage. Thank you for your attention.